Today we're going to be talking about glycanage and COVID-19, and we are joined by Gordan, Lauc and Daniel Bojar, and all of you will discuss the role of glycans in aging and COVID-19. I know from a few of you that are participating that a lot of you have already done research on this beforehand using the materials that were sent out. Great, so I'm hoping that we have a really good discussion. The discussion was initiated by Daniel Bojar, who's a 2020 Foresight Fellow in Health and Longevity, and he recently gave a Foresight presentation on the dark matter in biology and its role in COVID-19 that initiated this discussion. We're also really honored to be joined by Professor Gordan Lauc, whose extensive research really on glycans inter alia resulted in technology that is currently being used in GlycanAge. And so both of them will give a short presentation before we open up for a discussion with all of you. And Daniel will be up now, focusing on glycobiology, really to set the stage for Gordan's presentation that is focused on the state of the art, including potentially even a glimpse into previously unpublished results of his research. So that's exciting. Daniel, for now, is the 2020 Fellow in Health, Foresight Fellow in Health and Longevity, and he's a postdoctoral fellow at the Foresight Institute for Biologically Inspired Engineering at Harvard. He has harnessed the potency of cutting-edge machine learning to elucidate the biological functions of glycans since September 2019 already. And glycans are often described as the third language of life after DNA and proteins, and they're by far the most complex plastic biopolymer in humans. And Daniel thinks that those macromolecules are crucial to every human disease, but nevertheless, glycobiology is still kind of predominantly experimental and severely neglected. And so it's a really exciting area of research. And Daniel really was one of the first researchers to devise, develop, and apply that are derived from to glycans and begin to tame their astounding complexities. And so hopefully his research results in paving the way for exciting new insights and novel therapies for all of the grueling diseases that many of us are working to make less harmful. So I'm really excited to have you here, Daniel. Without further ado, I could just start with the presentation, and then we have Gordan coming up, and then we're going to open the discussion for all of you. I will do some in the chat for now, but the stage is Daniel. Welcome.
Thank you very much for this nice introduction, Alison. I will share my screen now. Okay. So I will keep it as short as I can so that we have enough room for discussion. And I will just give you a short overview of glycobiology and glycans in general, and then also how they relate to health in general and our work on that. And since I assume that at least some of you will probably have seen the salon, or at least are familiar with glycans in general, I try to keep the overlap as little as possible. So as Alison mentioned, my background by now is machine learning and glycobiology. So that's what I'm working on. But I start with a general introduction about glycobiology. And I thought, since most of these presentations start with the order of telling you what glycans are and then why they're relevant, or in what manner they're relevant, I thought to flip the script a bit and just tell you first what they do and why they're relevant, and then I will tell you what they are. So here you just see a human cell. It's the textbook depiction of a human cell. It's full of intracellular structures. It has a membrane, etc. We have tons of these in our body, of course, but actually how they look like is that. So these furry hairballs that are just covered with glycans on the outside. And this is not only true for human cells, this is true for every kind of cell. So it's very different from the textbook depiction. And if you go even on a smaller scale, on a protein scale, so this is actually from a very recent preprint on bioRxiv. This is the coronavirus spike protein in turquoise, and it's here. You see the cell membrane on the bottom, and it's a protein, of course. So you have these nice wiggly kind of structures here. But again, how it actually looks like is this. So this is, and the dark blue part are the glycans that cover the surface of the spike protein, and you can clearly see that most of the surface is covered by glycans. And even this picture is kind of erroneous because the cell membrane on the bottom is, or the lipid layer on the bottom is also glycosylated, right? So with glycolipids, etc. So this is even covered in a broader sense. And again, this is not only true for this protein, this is true for nearly all proteins. So it's maybe some of you know the movie They Live from in the 80s. It's directed by John Carpenter, and the plot is basically that the character finds a pair of sunglasses that when you put them on, then you see the actual meaning of things. So the subliminal messages in advertising and what they really mean. And I think glycobiology is like this pair of glasses that you put on, and then you see how biology actually functions and how it actually looks like. And I just
Daniel, may I ask a quick question?
Sure.
About this slide, with the illustration on the right with the blue glycans and polysaccharides, is that some kind of probabilistic thing, or is that really one single glycoprotein with all of its glycolytic hair?
It's a great question, and it's super relevant. And this is conformational averaging, basically. So you take multiple snapshots, and then you just depict all of the snapshots at once. So this is like the range of surface area that can be covered by these moving glycans, basically.
Yeah. So I mean, just to mention that other illustrations of the glycosylated spike protein will show a single configuration, and it's not quite so complicated as this.
Yeah, absolutely true. Yeah, but the great thing here is you can really see patches that will never or very rarely be covered with glycans, at least in the sample time frame, and that may be relevant for vulnerabilities in the protein. But I definitely agree. It depends what you show, how covered it will be in glycans. So glycans are actually huge in terms of numbers, and both in terms of mass. I mean, if you think about cellulose, for instance, cellulose is a glycan, and that's a lot of biomass on our planet, and chitin as well, and there are really tons of glycans. They're not only really heavy, they're also very diverse. So they are, as Alison already mentioned, they're the most diverse biological sequence that we are aware of, both in terms of composition, but also in terms of their structure, because they're actually non-linear in comparison to DNA or proteins. But I'll mention that later again. And then lastly, they're of course commercially super relevant. And just to pick a random class of therapeutics, in this case, monoclonal antibodies, which make up hundreds of billions of dollars in their market, they're heavily influenced in their function and their efficacy by the glycans that they carry on these proteins. So, I mean, it's probably an understatement to say that glycans are relevant for a lot of different areas, including human health. So to go a bit more into the molecular details of what glycans actually are, here are a couple of pictures of that. So these pictograms depict these chains of sugars, because that's what glycans are at the end of the day. So they're these sugar building blocks that you connect in different ways. They can be branched, like I said. And in humans, two of the major categories of glycans are N-glycans and O-glycans. So N-glycans cover at least over half to 2/3 of all proteins and modulate their structure, their function, their stability. Whereas O-glycans can also of course be attached to proteins, but what people mostly are interested in when they analyze O-glycans are the mucosal surface of our body. So the gut and other mucosal surfaces where they nurture, shape, and interact with the microbiome, and therefore have a lot of interesting functions. And of course also the blood groups are O-glycans as well. Just to go a bit more into even more detail in terms of diversity, to really appreciate that. So I told you that they are made up of these building blocks. These building blocks are called monosaccharides, and these are just funny-looking shapes and different colors and sizes, and then you can have different ways of how you can arrange them. And just if you do a very, very simple calculation of how many potential glycans you can get given the alphabet, then you see that the glycans are incredibly more diverse than DNA, RNA, or protein. Because for DNA and RNA, you have 4 potential.
Can you still hear Daniel? Is Daniel cut out for you too?
No.
Okay.
Potential options. For proteins, you have 20. Can you hear me now? Can you hear me?
If he turns off his video, his audio track might be easier.
Okay.
Yes.
I'll try that.
Perfect.
Well, but we love the slides.
Thank you. So basically, the idea is that glycans have the only alphabet of a biological sequence that is not universal. So every species has their own mixture of different monosaccharides that they can mix and match. But if you just think about the alphabet that is available overall, because that's what you could do in the lab at least, then even at the size of length 18, so a glycan of length 18 would have more potential configurations than we have atoms in the known universe. So that's a pretty big diversity, I think. Okay, yeah. So I don't want to steal too much thunder of Gordan's presentation later. So just a very brief outlook into how they modulate immune system function. Just a single slide here. So on the top left, you see antibody molecule, so the protein, and each copy of this protein has a single glycan attached to it. So for the immunoglobulin G molecule, and this specific glycan sequence that is attached to the antibody will define how effective it will be and in what way it will be effective. So it really takes the whole gamut from anti-inflammatory to pro-inflammatory, and that makes it also commercially interesting as well. So that's why there are companies that really take care of having a homogeneous distribution of glycans on their antibodies, and B, a specific kind of sequence there to modulate the functionality of the antibody. And the relations of function to the specific sequence motifs have been mapped out by decades of research, and Gordan's work really is really using a lot of that also to great effect, and he will talk about that later, I think. But I just wanted to tell you why this is important from a health perspective, just from a single aspect already. So that was sort of like a short overview of glycans and glycan biology as such, and then I'll just give you a couple of slides of what we're currently doing in the context of health with glycans. So we wanted to go about this from a machine learning perspective, just because of this huge diversity, of this huge nonlinearity, and of the lack of machine learning methods as to date for glycans, and to determine function from sequence in a glycan setting that could then help also in a health setting. So for that, we compiled this data set and then trained a recurrent neural network to learn sort of a language or at least patterns of glycans that you can then leverage in a predictive manner to predict then properties, functions, sequence motifs, etc. from glycan sequences. We did that for a couple of applications. I will not go into most of them. I'll only go into the immunogenicity. Just the other ones are basically predicting structural properties on the left, and on the right it's predicting whether a given bacterial strain is pathogenic purely from its glycans, because glycans have a hugely important role in most pathogenic reactions, and you can leverage that with our models. In the middle part, we try to predict if a given glycan is immunogenic based on its sequence, and immunogenic for humans specifically, because that obviously is dependent on the species, also dependent on the individual, on a lot of factors, but we get very good results in terms of that. And it doesn't really matter the details, what you see in the plots exactly, because I just use it as a segue here to what we actually do with that then. So the cool thing about a model like that is you get two things. You get, for the first thing is you get a representation of a glycan molecule that is learned by the model, and that is basically just a list of numbers that describes this glycan in some kind of multidimensional space, and that you can use to compare to glycans in terms of their similarity. And the second thing is you can predict a property, such as function or immunogenicity in this case. We use that for two things. So the first thing is xenotransplantation. So the idea of using organs that are grown in pigs and then transplanted to humans, and that obviously it doesn't work straight away because it's rejected as a transplant, and that's actually mostly the case because of the glycans on the organs of the pigs, because they are not immunocompatible with our immune system, and that's why people have started to generate these transgenic pigs, where they really basically switch off some of these enzymes that attach some of these sugar motifs, and we tried to predict based on the pig glycans which ones of them are predicted to be immunogenic, and those turn out to be the ones that people have actually targeted to remove them so that they get an easier transplantation, and we even get a few new motifs that could be the next barrier in this field. And then the second part here is we have this theory that a lot of the IBD type of diseases are at least in part mediated by inflammatory glycans that are produced by either microbiome members that are in disarray or by newly incoming pathogenic species. And one example has been shown in a recent publication last year, and it's a glycan depicted here, produced by Ruminococcusgnavus, that has been shown to be inflammatory in the setting of Crohn's disease. And if you put our model on that glycan, then you can then the model clearly predicts that this is an immunogenic glycan, and we think this could be a kind of screening tool, so to also pinpoint where are the problematic points in terms of gut diseases and inflammatory responses. And then just as a last slide here to sort of open the discussion, and then I'll hand over to Gordan for his part. So just there's a few papers that looked at the role of glycans in aging from a senescence perspective, and also in the last case, it's in terms of neural the clearing of debris and thereby function increase. And I think there are a lot of areas that are affected by this, especially as they are so plastic and can be rapidly changed depending on cellular conditions, that this is at least valuable for a biomarker, but could even potentially be used from a therapeutic angle if you could change the glycan patterns, of course, depending on the error of causality, as always. But with that, I just want to thank my collaborators that made our work possible, and I stop screen sharing so that I can turn over to Gordan.
Thank you so much, Daniel. This was fantastic. I think you brought everyone up to speed really nicely here. Gordan, thank you so much for joining. Thank you so much for making time. I'm super excited for your presentation, and I hope you can hit the ground running with Daniel's pre-work. For those of you who haven't read his bio, I'm going to share it again here in the chat. For those who would like to read up on it, there's a lot of content, I think, to digest. But for now, I think you're a professor in biochemistry, molecular biology at the University of Zagreb, with a lot of honorary positions, including one at King's College London and the University of Edinburgh. You founded a private research institute, Genos, which currently employs, I think, over 40 researchers that analyze the glycomes of over 150,000 people. One of your most prominent discoveries is the GlycanAge test of biological age, which is available from a company, and I'll share a few links to intro presentations in the chat. And 2 months ago, maybe now a little bit over that, David Sinclair and you wrote an editorial that suggested that biomarkers of biological age may be a better predictor of COVID-19 case severity than chronological age. And I'm really excited for exploring that thread a little bit more in the discussion. And then 2 years ago, I think you also established a global project named the Human Glycome Project, which is, I think, really exciting and aimed at understanding the structure and function of human glycans. And currently, I think, has over 300 investigators. So there's a bunch of threads, I think, that we can pursue in the discussion. For now, I don't want to take away any time from your presentation. Thank you so much for joining. I really appreciate you taking time and looking forward to the discussion.
Okay, thank you for the introduction. You clearly did your homework, so I don't have to repeat anything. I have a little bit of a storm around here, so if my internet breaks down, I will try to come back as quickly as possible. And it was a long day for me, so I'm a little bit tired. So if I'm too boring, please have some understanding. So I will tell you something about glycans and how do we think that they can be used as the biomarkers of biological age. But before I move to that, I will also give a little bit of introduction. I'll actually show the same image which Daniel showed, but I will start with the one on the left, which is something which director of NIH was blogging about. And this is the SARS spike protein without a single glycan. And that's a major mistake, which unfortunately many scientists still do. They look at this bare peptide part without these glycans, which indeed here are depicted as conformational dynamics. And the majority of proteins are glycosylated. And if you study a protein which looks like this, I think the best analogy is studying a bird which looks like this. So you can do a lot of research on a bird without a single feather. You can see it walk, you can see it jump, you can analyze its metabolism, but you will never see a bird flying. And I think it's the same thing with proteins. Without glycans, you will never understand the full level of protein complexity, because it's actually glycans which enabled the development of multicellular life, because the first RNA life was based on 4 blocks. Proteins are based on 20 building blocks. Glycoproteins have approximately 2,000 different building blocks, which can be combined and added to the polypeptide backbone. And it's estimated the glycoproteome is at least several orders of magnitude more complex than the proteome. And all this complexity is not directly encoded in the genome, because all our proteins are directly encoded in the genome. So if you know the sequence of a gene, you know the sequence of a protein. But glycosylation is a post-translational modification. It's a complex enzymatic process which results in some kind of a complex structures, which are then added to different parts of a protein. And this is partly heritable, but it's not heritable through a single gene, but it's heritable as a complex trait. So complex traits are usually things like, I don't know, height, intelligence, or anything which is complex, but glycans are chemical structures which are inherited as a complex trait. And through a series of GWAS papers we did in the last decade, we mapped, at least for IgG, there is more than 40 genes which form this complicated network, which decides about which glycan will these enzymes put. So these 4 are the enzymes which actually build a glycan, while these other proteins make a decision which glycan are we going to put in a given position to a given protein. And the problem with glycans is that first, structurally complicated, second, genetically complicated. So it's not easy to study them, and unfortunately, many people ignore them. And one of the first cycle is, of course, analytics, because if we cannot analyze glycans, we cannot work with them. And glycans are nonlinear, so you cannot just read the sequence. You have to do a complicated chemical analysis, multiple steps that you have to do before you actually put the glycan on a machine. And this is not easy to do in a high throughput manner. And actually, there are only 2 labs in the world which routinely do high throughput glycomics. One is my lab in Guinness, and the other one is in Leiden University Medical Center. Unfortunately, Nybert nearly ceased doing glycans after Pauline Rad retired. A little bit in Max Planck in Magdeburg. We were trying to start one lab in Beijing, but with Corona, this more or less stopped. And unfortunately, not even a single high throughput glycomic lab in US. Fortunately, now NIH has some kind of dedicated funding for development of glycomics. So I hope some of the US glycomic labs will also move to high throughput glycomics in a reasonable time frame. We are leading this field. We analyzed over 150,000 different samples in a lab, but not any samples, but samples coming from some of the best phenotype, genotype, clinical and epidemiological cohorts, including, for example, 20,000 twin samples from the Twins UK, number of longitudinal samples like the EPIC, SABRE, and so on. So we accumulated a huge amount of data, which we did in collaboration with some of the leading researchers all around the world, including the Harvard, Hopkins, Yale, Imperial, Edinburgh, and so on. And by combining the excellence of data which these colleagues collected with the technology for high throughput glycomics we developed, we actually enabled glycomics to move to mainstream, more or less. At least we were able now to publish clinical data in the leading journals, including the Circulation, Gastroenterology, Diabetes Care, and so on. So routinely, we now it's a situation that by adding glycomic data, our clinical colleagues can publish better than without the glycomic data. So I really like the change because until recently, it was not so easy to publish a good glycobiology paper. And one of the first discoveries we did in one of those huge studies that we did is that glycans change a lot with age. So this is an example of 4 cohorts. Each of them is over 1,000 people. We published it 6 or 7 years ago. And you can see that some glycans go up and some glycans go down with age in all those cohorts. Blue are men, red are women, and they all change in the same direction in all cohorts. And in principle, what we see that this type of glycans, which are the glycans which suppress inflammation, they go down with age, while these glycans, which are known to promote inflammation, go up with age. So as we are aging, our glycans are changing and they're not the same they used to be when we were young. And when we put this in a simple computer model, we were able to predict somebody's chronological age plus minus 9 years. So guessing a chronological age of plus minus 9 years is not so much fun. Epigenetics can do it within a year. But interesting thing here is that the difference between the glycan age we model and the chronological age is actually explained with a healthy and unhealthy lifestyle. And this is why when we publish this, editors publish an editorial saying, are glycans the holy grail of biomarkers of aging? So when we look into people and compare glycan age and the chronological age, we said that the difference is explained with the levels of insulin, fibrinogen, HbA1c, BMI, triglycerides, all those bad things we know that are not good to have. Also, when you look in the different patient cohorts, and we analyzed thousands and thousands of patients, we see that in many different diseases, and on this slide we have a lupus, ulcerative colitis, Crohn's disease, rheumatoid arthritis, Hashimoto, diabetes, colorectal cancer, the changes we see in the disease cohorts are the same as the effects of aging. So those pro-inflammatory glycans go up, anti-inflammatory glycans go down. So what we see here, we do not see a specific biomarker which would be specific for a disease, but we see some kind of a molecular change which is happening in many different diseases. And what we think is here, what we see here is a low-grade chronic inflammation and the effects of IgG glycans on balancing of the low-grade chronic inflammation. Because important thing about glycans is that they're not only biomarkers, they're actually functional effectors which regulate the immune system on multiple levels. And this is a good review a couple of years ago on the multiple levels where immunoglobulin glycans actually regulate the immune system. So we ask ourselves, okay, if this is really the case, if IgG glycans regulate the immune system on multiple levels, what will happen if people have a mutation which will affect their glycom and move it in a specific direction? And we did a huge Pliotropy analysis between the genes we identified in our GWAS studies, which we know now that they regulate IgG glycosylation, and checked whether there are known risk factors for some diseases or conditions. And we found that there are 94 different phenotypes where at least one SNP is shared between the IgG glycosylation and a phenotype. And some of them, like systemic lupus or Crohn or type 1 diabetes, would be something we would expect. But then we also observe Parkinson's disease or HDL or some dementias, which we would not expect to be associated with IgG glycosylation. And then we did a big analysis of everything which was published in the field. And this is a review with over 300 references showing that indeed in these diseases where we have this Pliotropy, glycans actually change in that direction. So this is actually fascinating to see that there is a mutation associated with the disease risk. You take a patient cohort and you see that glycans have moved in that direction. And then we went a step further and I asked all my colleagues all around the world, please collect me some samples in your region. And we collected 27 different populations with as wide age span as we could, and then compared how do IgG glycosylation change across the world. And we were more or less shocked actually when we saw this, that this is practically on a line with the expected lifespan and the development index of these countries. So if you live in a country like Papua New Guinea or Uganda, your glycans move toward the old glycans much faster than what would happen in England or Germany or some other developed countries. So it seems that this change of glycans from immunosuppressive to pro-inflammatory moves independent from the calendar ages, from the calendar year. It's some kind of a biological process which is driving this biological aging. And if this associates with expected lifespan, then we said, what is the first killer? And the first killer is cardiovascular diseases. So what happens with IgG glycans in cardiovascular diseases? So we observed in a series of studies we did mostly with our Chinese colleagues that people with hypertension have really altered glycans from the normals, but people with pre-hypertension already move in this direction. We also did a huge cohort of over 3,500 people in UK with Tim Spector and Jim Wilson. And we realized that the glycans we have today associate with the cardiovascular disease, the risk score, which is known risk score for cardiovascular events, suggesting that glycosylation today predicts future cardiovascular events and that people who would have more pro-inflammatory glycans will have cardiovascular events more frequently than people who have more immunosuppressive glycans or the lower GlycanAge. Then we went into one extremely valuable cohort, so-called EPIC study, They collected over 300,000 people in Europe. We got access to the 27,000 people which were collected in Germany approximately 30 years ago and followed for 10 years. So during the follow-up, there were 508 incident cardiovascular events, meaning heart attack or stroke. And then when we looked how predictive are glycans, glycans were as predictive as everything else we knew about these people, including the all known risk factors. And in women, the single glycan was more predictive than the entire AHA score. So this is extremely exciting because it says that glycans today are kind of predictive what will happen with your cardiovascular health in the future. And we are currently doing 2 large replication studies, one with Harvard with 3,000 people, over 1,000 incident events. This is already on the machines in my lab. And we have 5,000 samples coming from Freiburg. We'll do it towards the end of the year. So we'll see whether these 2 things will replicate in other cohorts. Because there is a possibility that this is actually causative. There is a study we did together with Phil Scholl from UT Southwestern, which was published in Circulation last year, where he showed in mice that if you have mice who have those bad glycans, they will develop hypertension. And if you take these glycans from one mouse and put it in another mouse, the other mouse will develop hypertension. And if you feed mice with a precursor of cellulation, the MANAC, which increases cellulation, and then these mice would have higher cellulation, you put them on a high-fat diet, they would become obese, but they would not develop hypertension. So in mice, artificial cellulation of IgG, which can be done with a dietary supplement, actually protects mice from hypertension. I was really enthusiastic that this could actually work in humans. So we found a cohort in NIH where people were actually feeding fed MANAC. And unfortunately, in healthy people, we do not see any effect. We still do not have a cohort of obese people who would be taking MANAC, so maybe it works only in obese people. But so far, it does seem in humans it's a little bit more complicated. You cannot just feed a MANAC. But we do see the same type of associations between the low salicylic acid and high blood pressure in humans. So generally, what is the general idea behind all this is that normally people go to see their physician when they're ill, when something hurts so much that they cannot normally function. So some of the organs is malfunctioning, then they get the diagnosis. And today, old people generally have chronic diseases which are never cured. They're only the symptoms are being treated. So if we find them early, maybe this can be reprogrammed. But if we find them very early, before any symptoms, then maybe a single or simple lifestyle change could change this wrong direction. And if we just can, for example, improve the IgG glycosylation and do not have this inflammatory driver, maybe the disease will never develop. And here we have a problem that we all know that if we live a healthy life, and this was a large study on I think over 100,000 people, which clearly showed that the more known risk factors you have a shorter healthy lifespan, and by living healthier, you can get 10 years of healthy life. But people don't do it. And people don't do it because it is difficult. For example, we know that caloric restriction works. These 2 monkeys are equally old. This one is on caloric restriction for I think 2 decades already. He definitely looks younger, but he does not look happier. And for people, it's not easy to make a decision now and to make a tough choice now to have a benefit in a few decades. So this simply does not work. So what we are hoping that if the test would give people faster feedback, it could motivate them to make changes sooner, live healthier, and actually prevent diseases. And the GlycanAge is something which can help people to do it because it gives an objective insight into the current state of low-grade chronic inflammation, although we cannot legally claim it because the GlycanAge is registered as an age test, because this would require this claim would require approval by the FDA and the European Medicinal Agency. And it is also a quantitative tool to evaluate effects of lifestyle intervention. So this is something which could give us a feedback. So we have a test that is reliable. We measure it quite reliably. It measures molecular effectors, so molecules that actually do damage. So these which promote inflammation and these which suppress inflammation. And this test actually integrates genetic information, epigenetic information, and direct environmental aspects of aging. And the key question is, can I change it? How can I change it? Because I think the key challenge for all biomarkers of aging is, can we change it? Can I do a healthy intervention and then change my glycans? And we don't have many people who tried interventions, especially for a longer time. One of them is me. I know my GlycanAge for 5 years, and it's lousy. I'm in my now late 70s, and it's getting worse. And it's my lifestyle. I'm still below 50 chronologically, so GlycanAge is horrible. And I tried different things after I learned this. I tried to sleep more, didn't work. I tried to be under less stress, it didn't work. I tried to exercise heavily, it didn't work. The only thing which works for me is diet. So for me, if I lose weight, my GlycanAge goes down. So I have to be this hungry monkey to have a low GlycanAge, which doesn't really work for me because I like to eat. But some other people might be luckier, maybe just more sleep would help, or a little bit less stress, or more activity would help. And we have some interesting examples. For example, this guy here is 76 years old chronologically. And he doesn't look like that. And his GlycanAge is in his 40s. So he's very happy with his GlycanAge. He claims to be a GlycanAge champion, but he lost his title to this guy over here, which is, he takes all possible and impossible supplements, but it worked. He's nearly 40 years younger than the chronological age. But some people can do it just by lifestyle. For example, this is my wife. She's nearly 30 years younger and she just lives a healthy life. I claim she's taking my GlycanAge because she's shouting on me all the time, but okay, I won't discuss that part. This is not science, this is anecdotal, but we are trying to do more science. We know, for example, that bariatric surgery works. If people undergo bariatric surgery, their GlycanAge generally improves more or less in everybody. Few cases it does not. And some people lose even 30 years within 6 months. Generally, obesity is one of the main drivers. This is a study of 2,000 twins in 3 time points over 20 years. And we see that glycans, that twins who are gaining weight are aging faster than twins who are losing weight. So by losing weight, your GlycanAge moves slower than if you keep your weight or if you gain your weight. The other thing we know that works is estrogen. So this is still unpublished. We are going to post it in a week or so, so it's not really confidential. But this was a study where menopause was chemical induced, and then women were given either placebo or the estrogen. So the estrogen replacement cohort stayed where they used to be. Placebo cohort increased by average 30% of their age. After the recovery, they all returned to the normal. So estrogen is something which also works. This test is available. People can go on a web shop, buy it, get it anywhere in the world, collect a few drops of blood, mail it back in the lab, and get results. Approaching the end of my talk, I would just like to compare glycans to epigenetics because people always ask me the same question. How does this compare to the epigenetic age? I am a big fan of epigenetics. We do a lot of research on epigenetics. And I think epigenetics is cool. And Steve's original clock is great. He started from 300,000 methylation sites, selected 353, and he's predicting age so accurately that it's actually not useful in large cohorts because the error is plus minus 1 year on average. So you can't do anything with that. Actually, now some countries are using it to determine the age of illegal immigrants. So Steve is now developing grim methylation clock, which is modeled to something else and not only the chronological age. I just saw a couple of days ago, there was another modeling of another clock. So there is a lot of information in epigenetics, but we still do not know to interpret it. And in particular, we do not know all those commercial tests, which were built to be a proxy for the Steve's original test. We really don't know what are they measuring. And I'm still waiting to see some real studies whether a specific test offered by a specific company actually associates with anything or not. On the other hand, for glycans, we are measuring molecular effectors, so molecules which actually do the work, they regulate the immune system. We know that they change in multiple diseases. We know that they affect these diseases, and that glycans also change up to a decade before people get ill. We know that anti-inflammatory glycans are the basis of immunosuppressive activity of IVIG, which is the registered therapy. We know that lifestyle interventions can change glycans, and we know that hormones can change glycans. I have a big research group. This is actually now a little more than half of it. I have to make a new photo. And my research is very generously funded by the European Union on multiple different levels. We invested over 15 million euros in the analysis of glycans, and this enabled us to generate all this interesting data. I have shown you just a little snapshot of what we have. And I thank you very much for your attention. And I will stop sharing the screen now so we can discuss.
Thank you so much. That was fantastic. Okay, we have a list of people that are lining up to ask you questions, if you may, so feel free to unmute yourselves at any point, and I'm going to stop your screen sharing if you don't mind. Okay, first one up, I think Rian, you had a question.
Yeah, Gordan, what an honor to sort of meet you, quote unquote, for real. I've followed your work for some time. I've read everything and watched videos. Anyway, super interesting. But the last slides that you presented, particularly the COVID stuff, is obviously new. And so that's really interesting to me because it seems, and I'm going to actually turn this into a question in a moment. If you've got the GlycanAge, roughly speaking, predicting COVID morbidity and mortality, and you got lifestyle predicting the GlycanAge, now we finally have kind of like a smoking gun for the extreme, connecting the extreme comorbidities with COVID and lifestyle. So I want to know your comments on that. Like, are you willing to kind of, on Chatham House rules, go on record and saying, clean up your lifestyle to avoid dying of COVID next winter? That's one question. Wait, one question. And then the second question is I got this from one of your papers, but I believe you were quoting someone else. And I just want you to reflect on this comment briefly for a moment. The quote was, the genome determines the possibilities of the phenotype, and the glycome is the phenotype.
That's not my sentence. Somebody else said it. I like it. Because if you think about this spike glycoprotein with all these sugars on the top, this is all encoded in the glycome, in the genome, but sugars are actually doing the work. Also on IgG, sugars are deciding to which Fc receptor will it bind. But let me go back to your initial question because it's much more interesting. So there is a part which I did not have time to talk about, and this is what we did in diabetes. So we see that with diet or with genes, you can change your glycans up to 10 years before you get to diabetes. And these glycans actually regulate the half-life of glucose receptors and the insulin receptor on the membrane. So glycan branching is a big part of a receptor presentation on the membrane, and it could also be the ACE2, which we don't know, but we are trying to establish. So what we believe that indeed a bad glycosylation, which could be a consequence of your lifestyle, and most probably because although I hate it because I like cakes, it's too much carbohydrates. So it's very likely that low carbohydrate diet would help in decreasing the the risk for COVID, but this is not something that we have evidence for. This is just, if you connect the dots, this would be my bet, which is personally for me horrible because I'm a carbohydrate addict. But it's, yeah, I would agree with you.
Thank you.
All right, great. We have a number of people, I think. Steve just said that his question was for the chat. Maria, do you want to unmute yourself next?
Yes, thank you. Well, really great talk. I have a quick question, and maybe I missed it. How promiscuous are glycans? Are these bad pro-inflammatory ones? Do they like to stick to something kind of more than the anti-inflammatory ones do? And are the changes in the levels of the good and the bad glycans, are they in any way reflective of the changes of the substrates that they kind of more preferentially
glycans are part of the covalent structure of a protein. So while protein is there, glycan is more or less there. Although there are some examples of extracellular reglycosylation, deglycosylation. So it's a part of a protein. It doesn't, it does not detach. It's not come and go. It's there. So it's made when the protein is made. So when we talk about IgG, and IgG has a very long half-life, couple of weeks. And when something changes in a person, it usually takes weeks before these glycans change. So IgG glycans, which are the basis for the GlycanAge, is something which actually changes very slowly. Only in some really acute infections, we see, sorry, my son in the background. Then we see glycans changing faster. But generally, these things move slowly and do not really react on a day-to-day changes in your diet or habits or whatever. Did I get the question right? No.
I'll have to, I guess I read a bit more, but thank you.
Thanks for the
question. Thank you so much. Okay, Cosmo, your question.
So I got a bunch of questions as sort of a follow-up from the talk I gave last week regarding the senescence-associated beta-galactosidase, which the cellular aging researchers seem to define as like the second most important aging biomarker of cellular senescence, is the activity of that one key enzyme. And that one key enzyme removes galactose units from glycosylation patterns on cells. And I can see from some of your prior work that most of the things that you focused on are the degalactosylation of these glycans. Would you say that that's pretty much the core pattern you're observing, is loss of galactose?
Yes, the lack of galactose, I would say the lack, not the loss, is something which is considered to be bad. Interestingly, there are a lot of enzymes which remove sugars, like the galactosidase or sialidase. We never saw these enzymes as a GWAS hits. So if people have a mutation in these enzymes, they do not pop out as kind of a regulatory mechanism. So I was thinking they would be more visible because, for example, there is an enzyme which adds sialic acid, which is sialyl transferase, and a single SNP in this enzyme, single nucleotide polymorphism, will explain 20% of variation in a population. In the same time, there is an enzyme which removes this sugar, and it has no effect. So the only answer I can give you is I don't know. So we haven't seen any evidence for the importance of beta-galactosidase for IgG glycosylation, but I cannot exclude it because the thing is also when something is extremely important, then there are not so many mutations there. So maybe we don't see it in a GWAS approach. So I can only say I don't know.
Well, one more question though. What about that ABO gene? So that ABO gene determines blood type. It's the second most important COVID-19 marker found so far, and it's like the fifth most important heart disease in GWAS studies marker. And the role of that ABO gene appears to be whether or not you get a galactose at the end of your blood antigens or a GlycanAge. Have you focused on blood group antigens in your work?
We encounter blood group antigens occasionally. Now we focus on IgG, which is not a blood group antigen, but I will completely agree with you. First, I think the blood groups evolved because we wanted to protect ourselves from pathogens. So we just stratify ourselves in 4 different subgroups, and many pathogens cannot just cross from one to another. And I think with the COVID, we see the same thing, with a blood group A being at higher risk and blood group 0 at a lower risk. And there was recently a paper showing that also spike glycoprotein binds to sialic acid. So it also has a lectin property. And there are a couple of, heparin also blocks it, which is also glycan. So there is a huge glycobiology part of COVID-19, which has been largely ignored. And the unfortunate thing with glycobiology is that there are so few labs which can take a couple of 100 or 1,000 samples that this is moving so slowly. So the first COVID paper on inter-individual variability in glycosylation will come from my lab probably. So the measurement will be finished next week. So we'll need a week or 2 to draft it. Then we'll post it somewhere. So, and I do expect that inter-individual difference in glycosylation would be very important.
Well, I'm putting together a mass spec lab if you want to collaborate. So that's the last thing I'll say.
Hey, if anybody on this group ever sees a new paper from Gordan, can you forward the link to the group?
Yeah, we definitely do that. And I think we have Carl and Amy now, and I want to make sure that we get to the prompt of how can we use COVID-19 as a specific impetus for accelerating health extension, and with this particular angle of glycans and the slide that Gordon presented on using them as a biomarker for aging. Okay, so Carl, you're next.
So the methylome clocks, age estimates based on methylation
Can you move
are
a little bit further away from the microphone. You're extremely loud. Thank you.
Methylome clocks are relatively stable, and people are using proteome and metabolome as age clocks, though those are less commercialized and published on, but lots of people are using them. And the metabolome clocks have a lot of the same kind of ability to predict specific diseases in advance with the certain patterns that you see. But the metabolome is more unstable in the sense that short-term things can perturb it greatly without necessarily seeming like they have a long-term, a big change to long-term death prediction. How stable is GlycanAge? So this is sort of how accurate it is, but how stable is it to short-term factors that might not necessarily. So HRV, for example, is sort of worse than all of those in terms of people use it as an age predictor, but it's very, very unstable to all kinds of things like temperature and short-term illness and things that don't seem to long-term matter.
So we did numerous studies to see how stable it is. So unless something happens, something really drastic and acute, like acute influenza, acute pneumonia, it changes extremely little with time. It's something like 1%, 2%, 3%. And this is also the level of our measurement error. So day-to-day stability is very large. And we also did a couple of months stability. So same person every few days, testing. It's more or less a flat line. So in women, it's a little bit more complicated because it is under estrogen influence. So the menstrual cycle does a little bit of oscillation. And for some time we were concerned about it, but it's also within a couple of percent, 2 or 3% would be the effect of menstrual cycle. So I would say this is extremely stable. So even frustratingly stable, because if you want to measure the intervention, you have to wait for 3 or 4 or 6 months to see that this measure is actually changing, which I think is good. Regarding the proteomics and metabolomics and epigenomics, they all have the same problem, is that they measure thousands and thousands of different variables and then model something. And then they model against what you model. So if you model chronological age, they will predict chronological age. And even if you move to another cohort, usually the same thing will not work. So the only thing which is really stable is Steve's epigenetic Lauc. So he really did it, it works perfectly. All these other are extremely interesting. I'm really following them to see how they develop, but I think they're still very difficult to use commercially because they are sensitive at so many levels. So proteome, if you look your plasma sample in the wrong way, you get a different proteome. Well, for glycans, we managed to develop an assay where you can dry a blood spot and mail it, and we can still get the same number. So it's, I cannot go around bragging about what I do, but I think it's the best test we have at the moment.
Thank you. Okay, then we have Amy, and then I want to get back to the prompt on how can we accelerate this and in the larger context of accelerating health extension.
Thanks, Gordon. The super interesting talk. Question: when you mentioned the diet seemed to be able to impact your glycolytic age and also bariatric surgery. Also, there's gradual changes over time. Also, some propensity to be associated with obesity. That screams gut microbiome and virome to me, especially in the sense that, because bariatric surgery and diet, and especially low-carb diet, are two of the things that are going to cut down microbial burden or inflammatory trends in microbiome virome over time associated with aging. So I was wondering if you have considered trying to collaborate with either a group just looking at microbiome and virome changes over time and the effect on these products, or also a company that's trying to innovate with microbiome-based therapeutics and see if you could get access to their data and see if you could see a change in the glycosylation when the therapeutic was administered. And I think virome, not just the bacterial organisms, but the viral organisms, which outnumber the bacterial organisms, could be having an impact on this, especially when you show me how just with one virus, COVID, you can see so many changes. Then the collective activity of the human virome possibly becoming more pro-inflammatory over time could be huge. And coming from a virome angle tied to the gut might actually increase interest in how COVID could influence virome and vice versa.
Okay, so microbiome is something we're extremely interested in.
Yeah.
We haven't done anything with the virome yet, but it sounds interesting. So for the microbiome, we have shown that if you do a fecal microbiome transplant, your glycans will change.
Interesting.
The second thing is, I think that the glycom, not GlycanAge, by the glycom, is heavily confounding the microbiome. And this is one of the bases why intermittent fasting actually works. Because when you don't do intermittent fasting, when you have all the time some food in your bowel, then all the aggressive bacteria can grow. Yes. When you do intermittent fasting, there is the soup they could eat. They can only feed on the glycom on your endothelium. And we co-evolved with those bacteria, and we know how to feed those which we like and ignore those which we don't like. So we collaborate a lot with Tim Spector. He has a lot of microbiome data. I'm extremely interested to collaborate with anybody doing different intervention studies like the probiotic study or anything which is well designed. We are more than interested to do this from our academic branch just to see how these things actually interact. But there is a big overlap of these two things.
Yeah, awesome, thanks.
Thank you so much. I know that we're already 3 minutes over time, but I would be super grateful, Gordan, maybe if you could share a little bit on what are your next steps in research. You mentioned that there's something that's coming out early next week, right? And then how do you have any tips or any proposals on how we can use COVID-19 to actually accelerate attention and funding in that area?
So we do something like 30,000 people a year from different cohorts, everything we can get our hands on. Currently, we have one is this COVID cohort. So if there is a COVID predictive biomarker in glycom, this would be extremely interesting. Another cohort we have at the moment is 1,000 people on 4 different diets for a year. So we'll see effects of different diets on the glycom. We are extremely interested in all intervention cohorts. We are organizing some intervention cohorts with with the different supplements. We have something like 700 mice from David Sinclair with everything he did. So there is a huge amount of things which can be done. Regarding the COVID, I think it's extremely important to keep the pressure on the government to fund the research in this metabolic health, healthy living, anti-aging, everything which can make people healthier, because what we know now is that COVID is more or less a common cold for 90% of people. 10% of people will get some kind of more serious symptoms, and something between 0.1 or 0.5 of them will eventually die. So there's only a subset of people who get a severe COVID, and we don't know to select them from. If we would have a biomarker and say, you are at high risk, go in a mountain, live in a cottage for the winter, this would be ideal, and then the world could continue going on. So I think what we can do, we can just go and be noisy, just showing the importance of healthy living, show that there is a biological age, it's not just chronological age. Because what was happening, for example, in Italy, and at some point also in Sweden, people were denied treatment based on age. They would say, you are too old. We cannot accept you to the hospital because our hospital is full. We are going to treat this 50-year-old guy who is so obese that he will die within the next couple of days. But still, the age was the primary criteria, which I think was a horrible thing to do. And this was never made official. So there were no official guidelines. You don't accept people above the age of 70. But this was happening. This was happening in Bergamo. This was happening in some other hospitals. And I think it's very important to avoid something like that happening in the future.
Okay, thank you. And very specifically, because most of your research is in Europe, right? So could you give like a little bit of perspective there? How hopeful are you that, let's say, GlycanAge could be like a predictor of COVID-19, right? So what would happen next there? Are you hopeful that you could get funding there? And if so, could this group help somehow?
So we have decent research funding. So GlycanAge as a company has a major problem that the glycans are completely, dark matter for most of most physicians, nearly everybody. So when somebody wants to, for example, buy a GlycanAge, then they would ask for an advice. Usually a friend or somebody who is in a medical profession, and they would say, I never heard of it, it's something stupid. So we have problems in promoting glycans as a field and a GlycanAge as a company. So GlycanAge is also looking for investors. I think we have Nicolina on a call who is much better than that. Nicolina? Yeah, she's here.
Okay.
Show yourself. So Nicolina is my daughter. She left home over 10 years ago went to London, became independent, and after being independent for 10 years, accepted to be a CEO of a company we founded together. So she's doing the business end. So if anybody is interested to collaborate or do any kind of commercial enterprise together, it's talking to Nicolina. If it's only science, it's talking to me, because if somebody starts talking to me about investing, selling, making money, I get bored. I'm a crazy scientist, but she's good in business.
All right, that's great. And Nicolina is in the health extension group now as well, if people want to follow up with her.
And she's based in London.
Okay, great. I'm wondering, Carl, you were, I guess, really interested in the topic of having different clocks and signal boosting them as biomarkers for aging due to COVID-19. Is there anything that's on your mind in terms of how we can use that for accelerated field of health extension more generally?
Well, I don't know about what you just said, Alison, but I'm very curious if GlycanAge is going to go in the direction of commercializing mouse versions of GlycanAge for research purposes, and also second independent question, whether or not you're going to try to come up with a combination methyloome glycanome. 10 years is just going to be too big of an error, but obviously there's a real signal there, and the real benefit's going to be when you have a combination that, where some statistical model takes all the data into account.
So two things. Mice, we are doing a lot on mice. The problem we have with mice is that it seems, it's not definite yet, so the jury is still out, but my gut feeling at the moment is that the mice which are grown in a pathogen-free environment, those specific pathogen-free environment, have much lower level of GlycanAge changes than the mice living with more interaction with the microbiome. So this is something we are currently exploring. We are having different mice in a different animal facilities. We did 100 plus mouse strains, and different mouse strains are extremely different in the way they glycosylate IgG. So not every mice is the same. And I think all the mice we have in our labs are immunological imbeciles. So it's what we saw is that actually strains which contain genes from the wild type mice had much more glycosylation than those, I call them immunological imbeciles. So extremely interested in mice, trying to find the best mouse model. Currently, as I said, we have something like 700 mouse samples from David Sinclair with all the interventions he did. So we'll see how it works. Regarding combination of methylation and GlycanAge, definitely interesting. We were thinking about it, and I was even very close to doing something together with a colleague of mine who has a company who is selling methylation, until I realized that he is selling only 6 CPG sites, which correlate 90% with Steve's clock, and nobody knows what is the 10% difference. So, his 10% variation could be a single thing, which is nothing to do with age. So we are trying to do something together, Steve, David, and I. And there are some ideas now that you really have to measure more than 5 or 6 CPG sites to get something reliable, which is then cost not effective because you have to run the entire EPIC assay. It's not a commercial product at the moment, but extremely interesting.
Well, have you seen them? Morgan Levine went to Elysium, and they claim their test is just $500 is 100,000 CPG sites. So
I believe this is doable. So,
but you definitely need a big you need a big bioinformatics machine learning model to make sense of that much data, which is what you would need to put in the Glycan data anyway.
So this is exactly what I said. So if you want to do the full, you cannot go below $500 to be at least a little bit profitable. We tried with the GlycanAge with a 600-pound level. It was not working. We had to go down. We are now around 300, which people are willing to pay. So I think with a $500 level, you don't reach so many people, and you cannot go down because Epic Array will never go down if you want to analyze 100,000 methylation sites. So there's definitely room for co-developing something. GlycanAge is just one of the aspects of aging. It's this immune aging. It's the low-grade chronic inflammation, which is giving you one set of information, which is useful. It's definitely not everything. Because what we also see, we see people who seem to have a healthy life with a bad GlycanAge. I know that my GlycanAge is bad because I live a horrible lifestyle. Work too much, eat too much. I know who is to blame for me, but I have some young guys in a lab who don't look so good. So there are definitely more aspects of aging than just the GlycanAge. There's no doubt in that.
You don't look so bad to me.
This is what I tell to myself. Yeah, we'll see what will happen in a couple of years. The thing which scares me is all the research we do show how highly bad GlycanAge correlates with the heart attack, with the stroke, with inflammatory diseases. And I do have some kind of inflammatory problems in my hands. So it's not that I'm not paying the cost of my lifestyle.
Well, look, if your own research, which you are the world's expert on, is scaring you, but not enough to do a low-carb diet, what does any of this even matter?
I do it. So I did it. So in the last 5 years, I lost over 10 kilos twice. And actually last time I was even keeping it for months. And then this COVID stupidity came in. And I was too close to the fridge whole day. And this killed me again. So I have to go on a diet again. But I'm trying to do low carb and I'm trying to do intermittent fasting. And I think this spring it was just too many fruits which destroyed me.
Well, remember what Yoda said, do or do not, there is no try.
Yeah, I know.
Okay, we're getting very philosophical here now. Okay, one thing that, could be an action item from this is, I know that a lot of people here are interested in exploring something like a kind of like private health commons, by which those people that are on different diets and do different tests and track different things share a little bit inside of this group and get the benefits out of that. So, one thing that we could be doing is getting the tests and just doing some studies, I guess, on this cohort for those who want to.
So something what we are always very interested is to learn more. And we often make things like, buy one, get 2 or 3 if you do an intervention and share the data. So it's not that we are just trying, I should be quiet because Nicolina is here. It's not that we are just trying to make too much money out of it. I'm trying to learn. That's my primary interest. Nicolina has to think about that she has to pay the salaries to the people who are employed. I'm not worrying that. So it's, but I think, Nina, we can go for 2 or 3 for 1 if they give us data, right?
Yeah, no, I think we're very keen to learn, and anybody who's willing to share their information, what they're doing, and we can find more things that work, it's absolutely worth doing that. So.
Foresight has a big network of people in other fields like nanotechnology. Gordan, maybe offline think about what underlying technological changes would speed up the cost effectiveness of the fundamental tests that are needed in this field. What's a 5-year long thing that would make this a lot cheaper, and how can the Foresight network help with reaching out to people who might be involved with that?
Yeah, we are thinking about it, and we tried many different things. So as Alison mentioned, I'm leading the Human Glycom project, and the Human Glycom project is talking to all of the big companies, including the instrument producers. And the problem is the moment we move to a cheaper or lighter version of analytics, we lose precision. And if we want to give people a feedback about their biological age, we cannot have a 5% error, because 5% error means the random moving up or down a couple of years, which then people would not understand, because even with intervention, we don't expect them to move for more than a couple of years in a few months. So we are currently stuck with this technology, which gives us a very low measurement error. Eventually, some kind of a nanofluidics with some kind of handheld devices would be ideal. But even for glucose measurement, the measurement error is too large. So it's I don't see it really quickly coming into the small portable devices. We are currently shifting our technology to all the DNA sequencers, the capillary sequencers, which are now retired for Sanger sequencing, and they can be used for glycan analysis. And we are trying to kitify the solution so the sample doesn't have to go back all the time to Zagreb, so we can have labs, at least a couple of them per continent. So we are moving in that direction. We are more than happy to collaborate. We still, for example, in US, we have very small footprint. We just collaborate with a small company, which is more or less shipping in and out for us. We are interested if somebody is willing to collaborate more in US, because I was warned by some of my friends, if you want to go for sure in US, it has to be a US company. You cannot come with European company in US and claim that you want to be big. So we are interested in many different things, but about these things, you talk to Nicolina, not to me. I'm a crazy.
Okay, I will leave an email in the chat box.
Can I pick your brain a little bit on the protocols, technology, and instrumentation that you primarily prefer to use? Because my background with glycan analysis is primarily via MALDI-TOF. We did a lot of that work back at the Mayo. But I understand you're using primarily HPLC for your approach, right?
So HPLC is the working horse at the moment because it's the most accurate. So MALDI is cool because you can do a huge amount of samples, but the measurement error is 5 to 10 times higher, especially for smaller glycans. So we were trying at one point to reduce it by doing multiple measurement of the same sample, but it's still, MALDI is good. It's good because you can do many samples. But personally, we do more HPLC. We do a lot of capillary electrophoresis recently because it's very similar. The problem is the attribution of individual structures is more difficult on CE. And we do a lot of LCMS when we go for a glycopeptide, which is also one of our focuses now. Go for a glycopeptide so that we are more confident that what we see actually comes from the specific protein. So now we have assays also for transferrin, fibrinogen, alpha-1 acid-glycoprotein, haptoglobin, many different things which we could do. So I think mass spec is probably the there's the largest perspective with mass spec just because the instrument producers are focusing there. And even just at the moment, the Waters is delivering a demo of their new BioAccure, which is actually, again, LCMS, kind of just MS detector. So it's, we can chat offline. This is probably not for this group.
Sure.
Okay, thank you so much for both Daniel and Gordan for coming. Thank you so much for staying over time. It was really a fantastic presentation. I think, many people here are quite eager to help in any way possible, and I'm going to try to get something on the self-experimentation side going with a subgroup of this chat. Thank you for joining. It was really fantastic. Next week, if we want to kind of like switch gears, we're going to talk a little bit about public policy advocacy, specifically based on the comment that Aubrey made in the introductory chat on, okay, who could we target right now and how, maybe sharing a few already successful narratives. Have we currently seen kind of like a revealed preference for actually caring for the elderly, or is this more like a kind of like enlightened self-interest or not so enlightened self-interest for staying home to save yourself, and like which are narratives that we could be pursuing and pushing in a public policy setting, and then even perhaps potential specific points that we could push for now. Are there any aspects of rejuvenation that we should push in addition for the immune system, which may become much more kind of like into the public eye now generally. So trying to talk a little bit about strategy there in the next session. That will be next Friday at 11 a.m. And thank you so much for joining. Thank you so much, Gordon and Daniel, for joining in your late time zones. It was really fantastic. I think people had a great time. Thank you so much.